In this video, we'll learn how gene expression is controlled in eukaryotes, specifically with respect to chromatin remodeling. The regulation of gene expression is more complex in eukaryotes than in prokaryotes. Differential gene expression is responsible for creating different cell types, arranging them into tissues, coordinating their activity, and forming the multicellular society we call an individual. There are different ways in which gene expression is regulated. Like prokaryotes, eukaryotes can control gene expression at the level of transcription, translation, and post-translation. Besides these, there are three additional levels of control that are unique to eukaryotes. These include chromatin remodeling, RNA processing, and regulation of mRNA lifespan or stability. In this video, we'll specifically look at chromatin remodeling as a way of controlling gene expression. In eukaryotes, DNA is wrapped around proteins to create a protein-DNA complex called chromatin. This prevents RNA polymerase from accessing DNA when it is tightly coiled within the nucleus. Chromatin remodeling involves DNA being released from its tight interactions with proteins near the promoter before transcription begins. Chromatin has a regular structure with several layers of organization. This organization is required to pack the extremely large DNA molecule, which consists of approximately 3 billion base pairs, into the tiny space inside the nucleus. The basic unit of chromatin structure is called the nucleosome. Several nucleosome units repeat throughout the length of the DNA molecule. Each nucleosome consists of the negatively charged DNA wrapped twice around 8 positively charged proteins called histones. One of these histone proteins, called H1, functions to maintain the structure of each nucleosome. Between each pair of nucleosomes, there is a linker stretch of DNA. The H1 histones may also interact with each other and with histones in other nucleosomes to form a more compacted structure called a 30-nanometer fiber. These 30-nanometer fibers come together to form higher-order structures. This elaborate level of organization allows DNA to be packaged in the nucleus and plays a key role in regulating gene expression. If you look at the different levels of chromatin structure, you can see that the DNA molecule is tightly wrapped around these histone proteins in units called nucleosomes. These nucleosome units repeat throughout the length of the DNA molecule. The histone protein H1 associates both with the linker DNA as well as the nucleosome to bring about stability within the structure. These nucleosomes come together to form higher order structures like the 30-nanometer fiber and subsequently the chromosome in its condensed form. This organization allows DNA to be packaged within the nucleus and also plays a key role in regulating gene expression. If you look at a micrograph of chromatin, you can see the string-like DNA wrapped around tiny knob-like structures made up of histone proteins. The proteins together with the DNA form nucleosomes. Initiation of transcription in eukaryotes begins when RNA polymerase binds to regions called promoters. But in order for this initiation to occur, chromatin must be relaxed or decondensed before RNA polymerase can bind to the promoter. So how is this tightly packed structure of chromatin altered in order to allow RNA polymerase access to genes that need to be expressed at particular time points inside the cell? Two major types of proteins are involved in modifying chromatin structure. ATP-dependent chromatin remodeling complexes reshape the structure of chromatin, whereas other enzymes that either acetylate or add acetyl groups, or methylate, add methyl groups to histones. Acetylation of histones is usually associated with the activation of genes. Methylation can be correlated with either activation or inactivation. The enzymes that add negatively charged acetyl groups to the positively charged lysine residues on histones are called histone acetyl transferases, or HATs. The acetylation of histones reduces the positive charge on the proteins, decondensing the chromatin and allowing gene expression. Other enzymes, called histone deacetylases or HDACs, remove the acetyl groups from histones, reversing the effects of acetylation and allowing the recondensation of chromatin. As we can see in this figure, the top panel is a micrograph of condensed chromatin, which can be decondensed by histone acetyl transferases that add acetyl groups on histone proteins within the nucleosome, leading to a more relaxed structure as shown in the bottom panel. This decondensed chromatin can be condensed again by the enzymes histone deacetylases. This concludes our section on chromatin remodeling. After going through this video, I'd like you to be able to respond to the questions listed on this slide. Thank you.